Hi everyone. Dear students, today let us study about the gametogenesis. Production of gametes is known as gametogenesis. We know that gametes are the sex cells which are important in sexual reproduction, and when a male and female gamete fuse together, a zygote will be formed and that can develop into a new individual. As because humans also reproduce only through the sexual reproduction, we also have the gametogenesis. Production of male gametes is what known as the spermatogenesis. Production of male gametes is called as spermatogenesis, and the production of female gametes is known as oogenesis. Whether it is spermatogenesis in males or oogenesis in female, the gametogenesis can be divided into three phases: multiplication phase, growth phase, and maturation phase. Now let us first take up spermatogenesis. Spermatogenesis starts or begins at the puberty, that is when a boy becomes adult, sexually matures. At that time, inside the testis in the seminiferous epithelial tubule, large number of spermatogonia are produced. Spermatogonia are the mother cells of the male gametes. They are diploid cells. They undergo mitotic division and increase their number. Now if you look at here in this array diagram, spermatogonia increase in number by repeated mitotic division, and this phase is called as multiplication phase. Out of the total number of spermatogonia produced, 50% of them only will further continue the process of spermatogenesis, but remaining 50% of the spermatogonia which are formed, they will be retained as the spermatogonia only. So as we see here, out of these two newly formed spermatogonia, one enters into the further process of spermatogenesis and the another remains as spermatogonium. So in this way, there is a renewal of, it is called as renewal of spermatogonia, so that all the time the spermatogonia are available in the testis to continue the production of the sperms. As we know that in human males, the production of spermatozoa or the sperms continue even at the age of 80 to 90 in old age. So the continuous supply of spermatogonia is possible through renewal of spermatogonia. Now let's see what happens further after the multiplication phase. The spermatogonium, which is destined to produce spermatozoa, undergoes growth phase. If you look at the growth phase, what is happening there? We just see there that the spermatogonium becomes larger, accumulates more cytoplasm, and it also undergoes another important process, that is DNA replication. The genetic material doubles up and each chromosome in the nucleus now becomes a double chromosome, and such a diploid cell only with doubled chromosomes. We know that diploid cells in humans have 46 chromosomes. Spermatogonium is also a diploid cell. Primary spermatocyte is also a diploid cell, but the difference is in primary spermatocyte, each chromosome has undergone duplication. Okay? Now this primary spermatocyte enters into the next phase, that is called as the Maturation phase. Let's see what happens in maturation phase. In maturation phase, as we see, the primary spermatocyte enters into the first meiotic division, and we know that first meiotic division is a reductional division. So the two daughter cells formed, which are called as secondary spermatocytes, they are now haploid because the number of chromosomes have been reduced. So if it is 46 in the primary spermatocyte, it will be 23 in the two spermatocytes, secondary spermatocytes. Okay, each of the secondary spermatocyte then undergoes second meiotic division, which is more or less similar to the mitosis. That means the number of chromosome further is not reduced, it will be remaining same. So we get 4 haploid cells, each having 23 chromosomes. 4 haploid cells are formed, and these haploid cells are called as spermatids. Now one thing you have to remember here, that when one secondary spermatocyte divides by second meiotic division, it is the separation of the chromatids, not the chromosomes. So from each chromosome, the 2 chromatids get separated, and as a result what happens is the resulting daughter cells will now have 23 chromatids. So these chromatids start behaving as the chromosomes. So that's how the mitotic division ends here, formation of the 4 spermatids from a single primary spermatocyte. Now the last part of the maturation phase is known as the spermiogenesis. In spermiogenesis, what happens is the spermatids, which are having round nuclei, and these round spermatids will change into highly motile, flagellated, tadpole-shaped spermatozoa. And this morphological change is called as spermiogenesis. Okay, that's about the spermatogenesis. Now let's have a look into how actually the spermiogenesis is taking place. We'll just go through the diagrams quickly. We see here that the round spermatid which has got a round nucleus, and acrosome vesicle has developed from the Golgi bodies that contains lytic enzymes. A flagellum has appeared and it has got 2 centrioles. This round spermatid will change into the elongated spermatid, as we see here. What happens here is the cytoplasm is pushed behind, the acrosomal cap becomes more larger, and the nucleus undergo compression, it becomes smaller, and mitochondria start getting arranged along the axial filament of the flagellum. This is flagellum. Okay, in the next step what happens is the large amount of cytoplasm from the gamete forming cell, that is the developing spermatozoon, what happens is the residual cytoplasm is separated. It gets collected out of the body of the sperm or spermatozoon, and we see that the further changes take place, and ultimately the male gamete or spermatozoon is formed, where we see that the residual body is separated completely, and now the sperm is fully formed. It has got a chromosomal cap at the tip of the head, then highly compressed nucleus, okay, which undergoes condensation, releasing water. It compresses, it becomes thin and short, and this part we take it as the head of the sperm, and then there is a narrow neck as we see there, and the two dots you see there, they are the centrioles. Okay, so these are the centrioles, these two. And then the other parts of the male gamete, if you take up the middle piece, we call this one the middle piece, which has got the spiral of mitochondria surrounding the axial filament. So that's providing the energy to the sperm for movement, because mitochondria are powerhouse of the cell, and that is the middle piece or engine room. And then the next part is highly elongated part, longest part of the spermatozoon, that is the principal piece, and the end one, at the end there is a tail piece. Tail piece, this is principal piece, and then this is the middle piece, which is also called as engine room, and head, and this is the neck. So that's how the spermatozoa are produced. Now just one idea about how this spermatogenesis is being regulated and controlled. So it is under the control of hormones. Mainly follicle stimulating hormone is responsible for the multiplication of the spermatogonia, as well as the mitotic division and meiotic division, ultimately ending up to the spermatids. But the spermiogenesis is controlled by testosterone, which is produced from the Leydig cells in the testes under the influence of LH or luteinizing hormone. Now let us take up the next process, that is oogenesis. Oogenesis is the production of the ova or the female gametes, and it occurs in the female human beings. And the main difference between spermatogenesis and oogenesis is that oogenesis begins in the embryonic stage of a female child. When the female child is still in the form of embryo in the mother's womb, we see that the oogenesis makes a beginning. The primordial germ cells, which arise from the extra embryonic membrane, enter into the tissue which is going to form the future ovary, and these primordial cells undergo the mitosis and produce large number of oogonia, and that is what multiplication phase. So during the embryonic stage itself, we see that around 2 million oogonia will be formed in each of the ovary of the fetus, and these oogonia get surrounded by one layer of the primordial germ cells, forming the primordial follicles or primary follicles. So by the birth of a female child, in her ovaries, in every or each ovary, we can say there are about already 2 million primordial follicles. But as the child grows, the number of follicles go on decreasing as they undergo degeneration, and by the time the child reaches a puberty, only 60,000 to 80,000 primordial follicles are retained. Now let's see how the oogenesis progresses further. So right at the childhood, after the birth of the female child, the when the maturation, growth phase begins, and during the growth phase, what happens is the oogonia enter into the first meiotic division. That is, first there will be a growth phase. Primary oocyte is formed just by enlargement, by accumulation of more amount of cytoplasm, and duplication of chromosomes takes place in the nucleus. So primary oocyte is formed, and that enters into first meiotic division. But this first meiotic division is arrested in prophase 1. That means maturation phase also has begun now, but it is arrested at prophase 1. Most probably in the diakinesis stage it will be arrested. And this arrest of the prophase 1 or first meiotic division will be there for 12 to 14 years till the girl child reaches a puberty. Only after reaching the puberty, the primary oocyte completes its first meiotic division to form the secondary oocyte. Now here one more difference you see, that this division of the primary oocyte by first meiotic division is unequal. One small polar body is produced, and another one is large secondary oocyte. So polar body retains very little cytoplasm, whereas secondary oocyte retains large amount of cytoplasm, and both these cells are haploid, as we know, because of the first meiotic division. And this secondary oocyte is now seen in the tertiary follicle or antral follicle. In the antral follicle stage, the completion of first meiotic division takes place. And further, the antral follicle will be growing into the Graafian follicle and ovulating. So the female gamete ovulates at the stage of secondary oocyte only. Okay. Now when the second meiotic division takes place, interestingly, second meiotic division takes place at the time of fertilization. As we see here, the sperm is entering from one side, and other side what happens is the nucleus divides, producing another second polar body with little cytoplasm, and now the ovum is completely formed. And that's how the oogenesis ends at the time of the fertilization. So that means out of 60 to 80,000 primordial follicles which are there in each ovary of the human female at the time of puberty, out of that only 400 follicles will reach the Graafian follicle stage and ovulate. And among the ovulated secondary oocyte, only few lucky ones which will develop into the children will complete the process of oogenesis, undergoing second meiotic division at the fertilization time. Okay, so that's about the oogenesis. Now let us have a look into the chromosomes and chromatids, taking up the both spermatogenesis, oogenesis, what happens there, how the chromosome number go on changing. Now if you take up in spermatogonia, or oogonia, the number of chromosomes is 46. And number of chromatids? No, because 8 the chromosomes have not undergone duplication. Okay. And what is the ploidy? The myocytes, that is the cells destined to undergo the process of gametogenesis or meiosis. So these cells, which are called as spermatogonia or oogonia, they are actually diploid. They are diploid cells. 2n we write for diploid cells. So ploidy is diploid. Next, let us take up the primary spermatocyte or primary oocyte So these cells are formed after the changes in the spermatogonium or oogonium. Those changes we have studied, that in the nucleus the chromosomes undergo duplication because the DNA is replicating, and therefore now the chromosomes are double chromosomes. So if you take up the ploidy of the primary spermatocyte or primary oocyte, the ploidy is again diploid only. Number of chromosomes 46 only, as because these 46 chromosomes have undergone duplication, so each one having 2 chromatids, so therefore number of chromatids will be 92. Clear? Then when these primary spermatocyte or primary oocyte further divide by first meiotic division, which is a reductional division, and they form the secondary spermatocyte or secondary oocyte, what we see there? The chromosome number reduces to half. Okay, 23 chromosomes are there now in each of these secondary spermatocyte or secondary oocyte. And what is the ploidy then? Therefore, the ploidy is haploid. These cells are haploid, and as because there are 23 chromosomes in each of the spermatocyte or oocyte, and these 23 are double chromosomes, so the number of the chromatids will be definitely 46, double of the 23. Whereas when the secondary spermatocyte divide by second meiotic division, okay, producing the 4 spermatids. So each spermatid also has got 23 chromosomes. And again, the ploidy is haploid. And how many number of chromatids are there? 23 only, because these 23 chromosomes what we take up, they are actually the chromatids. So from each of the chromosome in the secondary spermatocyte, for example, okay, when it divides, from each chromosome the 2 chromatids are pulled apart, and each one coming into one daughter cell. So that is how in the spermatids that 23 chromosome number is retained. Okay, and they are nothing but the chromatids which behave like chromosomes. Okay, now slightly let us have a light on how the variations are caused during the gametogenesis. Variation is very important part of the sexual reproduction. Okay, of course the sperm brings one set of the genetic content, the chromosomes, and the egg brings another set of chromosomes, and their combination gives rise to the variations. But before that also, during the formation of the gametes, what we see that we have actually 2 sets of chromosomes in our cells. One set we have got from the father's side, that is called as the paternal chromosome, and another set we have got from the mother's side, that is maternal chromosome. So these, now for easy understanding, only 2 chromosomes I have taken. These have undergone now duplication, as we see here, each one having 2 chromatids. Okay, and this is how in the primary spermatocyte or primary oocyte we find the chromosomes which are duplicated. And these 2 chromosomes, if you take up this one and this one, maternal and paternal, they have 2 chromatids, and their non-sister chromatids, the chromatids which are opposing to each other, they will undergo the crossing over. It's called as crossing over. And now what happens is, at the point of contact, the chromatin material or the chromosome breaks up, and the bits of the chromosomes are exchanged, as we see here. And that's how bringing about combination of the genetic content between these 2 chromosomes. And now we see that this cell has divided into 2 cells. So these 2 cells, these 2 daughter cells, now have one chromosome. Each one is double chromosome, of course. So it has become a haploid cell now. Okay, just like the secondary spermatocyte, for example. And further, when these 2 cells undergo second meiotic division, from each of the chromosome, the 2 chromatids are separated, and they enter into separate daughter cell. So from this cell we see that these 2 we can say spermatids are formed. So these 2 chromatids have got separated and they have come to lie in these cells. So likewise another cell also divide, producing 2 more cells. So these are the spermatids we can take up, and all these are haploid, and each one will be developing into a spermatozoan. So we see that out of 4, 2 cells retain the original genetic content, one of the paternal side and another of the maternal side, and the 2 are the recombinant type of cells where there is a exchange of chromosomal bits. Okay, now let us quickly go through the comparison between spermatogenesis and oogenesis. It will make you get this topic revised. Okay, now if you take up a comparison between these 2, few similarities but more differences you have. So spermatogenesis occurs in testes, oogenesis occurs in the ovaries. Spermatogenesis begins at puberty, oogenesis begins at embryonic stage, right? And the spermatogenesis has got 4 phases, rather 3 phases we should say. That is the first one, multiplication phase, then growth phase, and maturation phase. So this should be 3, not 4. So 3 phases we find in both, and then that is a similarity. And then if you take up the other points here, a single spermatocyte gives rise to 4 spermatozoa, whereas a single oocyte gives rise to only 1 ovum. Okay, single oocyte gives rise to only 1 ovum, because we have seen that another cell will be at first meiotic division, 2 cells produced, 1 will remain as the secondary oocyte, and another is a polar body, isn't it? Similarly, when secondary oocyte completes second meiotic division, we see that another polar body is formed and only 1 ovum or egg is formed. But in case of the spermatogenesis, 4 spermatozoa from 1 primary oocyte. Now 200 millions of spermatozoa are produced on the basis of every day, you can say, in adult human male, whereas in adult human female only 1 ovum, sometimes 2, will be produced per month. Okay, usually one ovary produces the ovum or ovulates this month, and next month another ovary will do it, but rarely, sometimes both the ovaries produce the ova or egg, so rarely two, but usually one ovum per month. Okay, so lot of difference in the numbers. And then spermatogenesis continues even in the old age. At the age of 80 or 90 also, the sperm production continues, whereas the oogenesis stops at the menopausal age, that is 45 to 55. Then prophase of first meiotic division is very short. Prophase of first meiotic division in spermatogenesis is short, whereas prophase of the first meiotic division is very, very long in case of the oogenesis, as we see that till the girl child reaches the puberty. So for about 12 to 13 years, the prophase 1 will be arrested. Then second meiotic division completes during spermatogenesis only in case of spermatogenesis, whereas second meiotic division completes in oogenesis only at the time of fertilization, as we have noted earlier. Okay, and next, spermatid further undergoes morphological change to form the sperm, that is what called as spermiogenesis, but there is no such change in the otid or in the ovum. Okay, then in case of spermatogenesis, the hormones which regulate are the FSH and LH via testosterone. As I already told you, the spermiogenesis is controlled by testosterone, whereas the remaining other part of the spermatogenesis, right from the spermatogonia to spermatid, that is controlled by FSH. Likewise, in the oogenesis also, FSH and LH are the main hormones which are helping in follicular growth and ovulation. So with this, we have come to an end of this topic. Dear students, keep studying, keep in touch with your subject. I will meet you in the next lecture. Bye bye.